last talk. I understand I'm the only one between you and lunch. So I'm going to talk about our team's effort on asking the central question on how do environmentally and clinically important bacteria can respire without oxygen-like soluble molecules. And I'm Nikhil, and both my team member Olivera and Carlos are here, so please say hi. And what we are focusing on is a process called extracellular electron transfer, or a EET for short, where the structures which we call protein nanowires, these are hair-like filaments on the surface of the bacteria, and they serve as a nanowire for this bacteria to transport electrons from inside to the outside. And these wires contain a stacked heme molecule, and this heme molecule, which I'm showing in the bottom, allows electrons to move at ultra-fast rate over 100 femtosecond across distances 10,000 times the size of a microbe. So with the support from HFSP, we were able to focus on these two cytochromes called outer membrane cytochrome S and Z, and we were able to identify the machine which makes one of these wires called OMCS, where we identified the gene cluster, and using genetics and structural biology, we were able to identify some of the components involved in this machine. And we were also able to develop a new imaging method. We were able to directly image electrons produced during metabolism by a technique called electrostatic force microscopy. And here we put bacteria on a graphene oxide, and as it reduces, it changes color, and we can map that color, which becomes white because of this electron transfer, and we can quantify this color about how each wire is transferring electron. And a big surprise there was that this PilA protein filament showed a very low activity, and the reason it was surprised because since 2002 it was thought that PilA are the wires which are transferring the electron, because genetic and many other studies had showed that if you delete this PilA gene, bacteria just cannot grow or form filament. But we found that there is a completely different protein called PilA-C, shown here in the orange blue color, which covers this hydrophobic PilA-N protein shown in orange. So they just cannot even assemble together. And they also have a very low electron conductivity. So when we measure electron current through this PilA filament, it's very, very low. So they cannot act as a nanowire, and they're also located intracellularly. So the question here is what exactly these PilA are doing, and with our HFSP team, we're able to discover, and can you please click on the video, that this PilA filament actually act as a piston, and they are involved in secreting the nanowires. And yeah, thanks for the video. So using genetics, we initially showed that the PilA deletion inhibits secretion. If you complement the gene, restores. So that explained all the prior genetic data. We were able to show that these PilA are really acting as a piston to push these cytochromes out. And then our team was able to use this hybrid system to discover that how are they moving electron. And with that, I want to end by showing what are the next applications possible with this wire, and we are focusing on some of these applications. And I want to end with this quote that it's all about electrons, and life is nothing but an electron looking for a place to rest, by St. George. Thank you very much.
Thank you. Thank you. And